Okay, so welcome again to the next presentation. I hope you enjoyed today's lecture. Now we will move the focus onto how we use microsensors in microbiology and within biomedical sciences and also within renewable energy research. So I will just share my presentation here. Okay, great. Yeah, and this is the second lecture of the day. Tomorrow we will go to the lab and do the live demonstrations of the microrespiration system and the microprofiling system. Just a little bit about me. My name is Daugaard and I am product manager here at Unisense. I have a PhD in nanoscience from Aarhus University, and I have been with Unisense for around 6 years now. You are always welcome to contact me by email, and I will also be happy to connect with you on LinkedIn. Yeah, this is the outline for today. First, I will briefly introduce you to the Unisense microsensors and explain why microsensors can be a nice tool for analyzing biological samples. Then I will go through some of the things you should consider when you are preparing your microsensor setup. And this will be followed by an overview of the different microsensor systems. Then we will explore some examples of microsensor applications within microbiology, cell studies, cancer research, neuroscience, and also renewable energy research. And at the end, we will have time for your questions. So, Unisense sell microsensors that can measure the different analytes that you see here. Lars has already told you about the different type of sensors and how they work. He did that yesterday. The tip of the sensors we can make down to less than 10 micrometer, and all the microsensors can be used to measure both in gas phase and in liquid. The microsensors must be connected to an amplifier to amplify the small signals from the microsensors. And we sell single-channel amplifiers and multi-channel amplifiers. To the left here, you see a single-channel amplifier having one channel for hydrogen and also a temperature channel. To the right, you see the multi-channel amplifier that has several channels, and that also means that you can measure with different types of sensors at the same time. So the choice really depends on how many sensors you would like to measure with. All the uniamp amplifiers, they allow for temperature compensation when you use a temperature sensor. All the sensors need to be calibrated, and a good calibration is really crucial for getting good results. The gas sensors, they respond to partial pressure, both in the gas phase or dissolved gas in liquid. And you must calibrate the sensor to relate the sensor signal to a concentration unit. Calibration is done in our software, and you can choose different units in the software. Could be concentration units, for example, micromole per liter or milligram per liter. And you can also choose partial pressure units like Pascal and millimeters of mercury, which is often used to express the partial pressure of oxygen in blood and tissue. It is important that you calibrate at the same temperature and salinity that your sample has. So if you have a biological sample at 37 degrees, you should also do the calibration at 37 degrees. The response of the sensor is linear, and therefore a 2-point calibration is sufficient. Here is an example of an oxygen calibration curve where we have a 0 point where we measure it in a solution without oxygen, and then we have a known concentration of oxygen up here. In the software, you find this concentration table, and it calculates the concentration of the desired gas at a certain temperature, salinity, and pressure. And we always recommend that you do the calibration before you start a new experiment. We sell calibration kits for oxygen, nitrous oxide, hydrogen sulfide, pH, and redox. And the calibration kits, they contain the reagents for the calibration. They also contain this calibration cap that makes it very easy to perform the calibration. So why should you use microsensors? First of all, you will get real-time measurements. This means that you can follow the sensor signal all the time. Because of the small tip size of the sensors, you get a non-destructive or a minimally invasive measurement, and you do not disturb the chemical gradients in your sample. With the small tip size, you can obtain a high spatial resolution. A rule of thumb is that the spatial resolution equals the tip size. So this means that if you're using an oxygen microsensor with a tip size of 50 micrometer, you can get independent measurements at a distance of 50 micrometer or larger. The small tip sizes also allow for a fast response time, and the microsensors have a low analyte consumption and also a low sensitivity to stirring. And why is this an advantage? You can characterize microenvironments. It could be a biofilm or a tumor microenvironment. You can document the heterogeneity of the biological systems on the microscale. You can measure directly in tissue and organs, both ex vivo and in vivo in living animals. You can do live cell analysis. You can look at metabolic rates, production and consumption rates, and you can measure in very small volumes. So choosing the correct tip size is very important for your measurement, and the choice of tip size depends on the sample and the type of measurement that you would like to do. A small diameter has a number of benefits. It gives a high profile resolution. It disturbs the sample very little, and it does not block diffusion, and you can obtain a very fast response time. But a small tip is also fragile, and this must be considered when you prepare your experiment. To the right here, you can see that increasing the tip diameter by a factor of 10 increases the area of the tip by a factor of 100. So this could have an effect on your sample, and it's really important that you choose the right tip size. Here are some general tip size recommendations, but it always depends on your exact application, which tip size to choose. And you're always welcome to ask us for advice regarding this. For biofilm and colonies, we typically see that people use very small tips to get a high resolution profile. The tip depends on the thickness of the biofilm and also how many measurement points that you would like to do throughout the sample. Tissue of live animals. For soft tissue, a very small tip size is also typically used to be minimal invasive in the sample. But for some more rigid tissues, we recommend larger tip sizes. And if you have some very rigid tissue, we recommend that you make a hole first with a needle. For cell studies, a small sensor tip is also typically used to get high resolution, both for cells growing in monolayers and when measuring inside 3D cell cultures. Small volumes depends on the volume, but we can measure very small droplets with the microsensors. For microrespiration measurements, you should use the microrespiration sensors that are designed specifically for the microrespiration system, and I will come back to this type of measurement later. Bulk measurements, when you are going to measure in a larger volume, maybe in a closed system, you do not need the high resolution from a small sensor tip, and you can use the larger sizes. It could also be that you needed a sensor that is mounted in a configuration like in a needle or in a steel tube or in a flow cell, so that it can be integrated with your setup. Yeah, so when preparing a setup, there are some things to consider. First of all, you should think about, would you like to do a profile, or is it a bulk measurement that you would like to do? If you would like to make a profile, what spatial resolution do I need? And here we have the rule of thumb that the tip size equals the spatial resolution. How do I obtain in situ conditions? You could think of flow, temperature, salinity, and so on. And very important, how do I protect the sensor tip from breaking? It's important that you fix both the sensor and the sample, and you can use a micro manipulator like this to mount the sensor and position it. How do I introduce the sensor? Should I use the micro manipulator here, or do I need to pierce a septum, or should I mount it in a compression fitting? These things you also have to think about. How do I ensure a steady state in my system? And how do I also ensure stable and well-stirred conditions? Finally, it can be a good idea to consider using a stereo microscope to have a look at the sensor tip when you position it on the sample. And I will come back to this option later in the presentation. Okay, then there are some special considerations when you are going to measure in tissue. First of all, disturbance. A smaller tip size gives less disturbance, and disturbance could be deformation of cells. It could be blocking of diffusion of nutrients and so on. So a smaller tip gives less disturbance. Tissue compression. Tissue may be compressed in front of the tip when the sensor tip is inserted into the tissue. And when the tissue is compressed, the blood flow is also compromised, and this means that the available oxygen is reduced. And this you will observe as a relatively slow decrease of the signal. And the oxygen signal can finally reach zero. A way to work around this is that you insert the sensor into the tissue a bit too far and then retract the sensor again to a point where you can see that the sensor signal is stable. And this effect is most pronounced with large tip. We can also make the sensor tip more slender and more sharp so that we reduce the pressure on the tissue. If you have to go through skin, it can be a good idea to cut it open first or pierce with a needle before inserting the sensor into the tissue. Obstacles. Some tissues can be very hard to penetrate. We know that the kidney capsule can be very hard, and therefore it can also be a good idea to cut a hole or pierce with a needle before inserting the sensor. Then it's very important that both the sensor and the samples is fixed. The sensor tip can break upon lateral movements, so it's very important that you fix both the sample and the sensor. And if you're working with a live animal, you have to make sure that the animal doesn't move too much. Yeah. So now I will go through different microsensor setups and ways of mounting the microsensors. We sell a complete system for making microprofiles called the microprofiling system. This system holds and positions the microsensor, and it can be operated both manually by hand or automatically by the software. When operated automatically, the depth and the step size, the measuring time, and the number of replicates for the experiments, they are all managed in the software called profiling, which is part of our sensor trace suite software package. Here you can see a microsensor moving towards a bacterial colony growing on an agar plate. Yeah. And here you can see what you get from it. You will have the depth position here on the vertical axis, and you will then get the analyte concentration on the horizontal axis. So you get this nice profile throughout the sample that you're looking at. From the measured profiles, you can calculate the production and the consumption rates at the different layers. And this is done in the profiling software, where you can calculate the production and consumption rates based on a model calculation of the measured profiles. Non-linear gradients here indicates production or consumption. My colleague Tae will demonstrate how to measure the profiles and also how to analyze them tomorrow when he's doing the live presentation in the lab. Okay, it can be a good idea to visualize the sensor tip on your sample, and we sell this side stereo microscope that is optimized for microsensor use. And this will allow you to very precisely position the sensor on the desired spot on your sample. The microscope integrates with our software, and you can take pictures at different depths, and the pictures will then be saved together with the depth position of the sensor and the sensor measurement. The picture to the right here shows a sensor tip taken with the microscope on a surface of an orange. Okay, so in the next system that I will introduce here is the microrespiration system, and it can be used to measure production and consumption rates in closed systems. This could be rates of microbial suspensions, cell cultures, algae, small animals, small eggs, and so on. The system is compatible with all the Unisense microsensors, and you can measure one or two analytes in your sample. The sensors for this system is mounted in this blue aluminum guide with a plastic cap that fits into the lid of the microrespiration chambers. And the cap here ensures correct insertion through a very narrow capillary hole. And the diffusion in and out of the chamber here is so slow that we can consider it closed. The rack of the microrespiration system can hold up to 8 chambers, and you add a glass magnet to each chamber, and the stirring is then controlled here by this stirrer controller. It is also possible to inject the substance into the sample. It could be an enzyme or drug or inhibitor or substrate, and you can then measure the real-time change in metabolism. Yeah. We sell glass chambers with sizes from 400 microliter and up to 400 milliliter. And you can see the different chambers and the dimensions of them in the microrespiration system manual. Here you can see the double chamber that is used for measuring with 2 sensors in one sample. And over here you can see the injection lid that is used when you are going to inject a substance into your sample. And to the right here, you can see how you can protect your sample. If you are looking at a small animal or a small egg, you can put it on top of a metal net here to protect it from the stirring from the magnet. I will just briefly introduce you to another system called the nanorespiration system, and that system is used for measuring extremely low respiration rates of small organisms. And with this system, you can measure rates on single individuals, such as small eggs, oocytes, and embryos. And the measurement is done by positioning the organism in a small well, and then you measure the gradient down towards it. And the oxygen consumption rate is then determined from the slope of the profile you measure. The system consists of a small rosette with small glass capillaries where the samples is placed. And the microsensor is then positioned on the micromanipulator as shown here on the photo. If you plan to use the microsensors in setups where the sensor should be mounted directly in your system, we have different mounting options available. This could be relevant if you plan to measure in small cuvettes, in culture flasks, in fermenters, in bioreactors, in photoelectrochemical cells, and in flow systems. Then we have different options, and you can see some of the configurations here. We have different types of needles. We can also put the sensors into different steel tubes. And we also have these different types of flow cells that allows you to measure in line. Okay, now we will explore some different examples of studies using microsensors within microbiology, biomedicine, and renewable energy research. So the first examples are within microbiology, and I will show you how you can measure both in bacterial cultures, in colonies, and in biofilms with the microsensors. The first example here is about hydrogen production from a cyanobacterial mutant. This study uses the microrespiration system to look at the hydrogen production. The authors here used the double chambers, so they inserted a hydrogen sensor and an oxygen sensor into the same sample here. And then they could measure the hydrogen production and at the same time look at the respiration rate from the bacteria. The authors created this cyanobacterial mutant where they fused the hydrogenase to its photosystem 1. And on the drawing down here, you can see how the electrons go from the photosynthetic train to the hydrogenase up here where the hydrogen is produced. Here you can see the hydrogen data showing that the cyanobacterial mutant produced up to 500 micromolar hydrogen under anaerobic conditions in continuous light. The authors could also show that the hydrogen produced was not taken up again and consumed in the bacteria, which was the case for the wild type. So they could really show a sustained hydrogen production in the bacteria. Here is another study looking at hydrogen production, and this time from algae. In this study, the sensors showed continuous hydrogen production using acetate-enriched wastewater. And the hydrogen sensor here was mounted in their own reactors. So they used the hydrogen sensor mounted in a needle for piercing. So they pierced a thin rubber septum here on top of the reactor, and they could then measure the real-time hydrogen production. The data is shown here. They measured for up to 15 days in 2 different algae species, and they could then see this sustained hydrogen production when they illuminated the sample with light. Okay, so in the next example here, the researchers grew Pseudomonas aeruginosa colony biofilms on plates. You can see them over here as these red spots. And they used the oxygen sensor and the redox electrode with a tip size of 25 micrometer to make microprofiles in these colony biofilms. The authors studied the microenvironment and the reduction of phenocines, which are a type of antibiotics and an electron acceptor produced inside of the bacteria. And they made a mutant where the phenocine production is depleted, and they compared these 2 strains. So you can see the profiles here. You could then see that for the oxygen profile, it looked the same in the wild type and in the mutant. But when we looked at the redox potential, it decreased with depth in the wild type, but stayed the same in the mutant. So this decrease showed that these phenocines contribute to balancing the redox state under the hypoxic conditions. So this is important knowledge about the microenvironment in these colony biofilms. Okay, so this is another study. The authors here looked at cystic fibrosis, and chronic infection is one of the complications in cystic fibrosis because Pseudomonas aeruginosa biofilms form in the lungs of cystic fibrosis patients. The researchers here, they collected sputum samples from cystic fibrosis patients and then placed them in these glass vials. And they then made oxygen and nitrous oxide microprofiles throughout the sputum samples. You can see here that there was a lower anoxic zone and that nitrous oxide started to get produced. Nitrous oxide is an intermediate in the denitrification pathway, and these results indicated that the bacteria may require energy for growth by denitrification when oxygen is absent. Yeah. Okay. So now I will show you how you can measure in cells with different setups. This study uses the microrespiration system to look at the oxygen consumption rate in cancer cells. They used the MR system, as you can see here, and the oxygen sensor, and they looked at the oxygen consumption rate in a breast cancer cell line. They then used this injection light and they injected substrates or inhibitors, and they could then see how that affected the oxygen consumption rate. So you can see how the substrate increases the oxygen consumption and how the inhibitor here slows down the oxygen consumption rate. The authors also injected different anti-cancer drugs, here among curcumin, and they evaluated the effect on the cell metabolism. They found that treatment with curcumin induces an energetic impairment in the cancer cells by inhibiting ATP synthase activity and also by decreasing ATP production and oxygen consumption in different cell lines. Spheroids and organoids, which are 3D cell models, are getting more and more attention as cell culturing models because they mimic in vivo conditions. Mesenchymal stem cells, they are under widespread investigation for use in regenerative therapies, and it has been demonstrated that these cells have an increased overall function and an improved survival when they are grown in these 3D structures. So the study here investigated the oxygen concentration profile in the spheroids to evaluate if they had a hypoxic core inside. So the authors made 3 different sizes of spheroids, and they then measured the oxygen concentration all the way through. They found that there was no hypoxic core in these spheroids, even for the largest ones. And this really increases their potential for clinical translation. You can also study the oxygen conditions of in vitro cell cultures with monolayers of cells. The respiration rate of the cell culture combined with the low rate of oxygen diffusion in the medium, it leads to a decrease of oxygen tension close to the cells. And the study here described these gradients using the microprofiling system, and they used an oxygen sensor with a 10 micrometer tip. They studied a human breast cancer cell line, and they inserted the sensor here with an angle to avoid breaking the tip if it hits the bottom of the flask. So the sensor tip can flex a little bit if it hits the bottom. They could see here a decrease of oxygen when going from the gas phase of 4% oxygen and then going down through the medium down to the cell layer. So you can really see here that there is a difference in the oxygen concentration that the cells experience and the oxygen concentration in your incubator. And this is, of course, important when you relate parameters to the conditions that the cells are actually experiencing. Okay, now I will show you 2 studies that uses the microsensors to measure in tumors in vivo. This is a study where the researchers used the oxygen microsensor to measure inside the tumors of mice in vivo. And in this study, the researchers looked at these perfluorocarbon nanoparticles to enhance the efficiency of hypoxia-based drugs. Tumors, they have this hypoxic core with a very low oxygen concentration, and this environment is a target for some drugs. In order to reach the tumor site and the hypoxic core, the tumor must also be penetratable. And these nanoparticles, they both enhance the tumor permeability, and they also led to long-term hypoxia. So the authors placed an oxygen microsensor inside of the tumor, and they then injected locally these nanoparticles. They could then monitor the real-time changes of oxygen inside the tumor after the injection of the nanoparticles, and they could see that the perfluorocarbon nanoparticles, they decreased the oxygen inside by 80%. Another study here used the pH microelectrode to study the acidic microenvironment of tumors. The acidic environment facilitates tumor expansion and metastasis, and it is therefore a hallmark of solid cancer tissue. It has been proposed that an alkaline diet may reduce the risk of cancer development. So in this study, the authors investigated the consequences of giving oral sodium bicarbonate therapy during breast carcinogenesis. As the authors moved the pH microelectrode deeper into the tumor, the pH decreased, and when they moved the pH electrode to the outer regions, the pH increased. They moved the sensor inside the tumor 2 millimeters at the time, and they placed the reference electrode in the interperitoneal space. They found that the oral administration of sodium bicarbonate increased the pH inside the tumor, but they did not have an overall effect on the breast cancer development and the tumor growth. Okay, so now I will turn to neuroscience, where especially the oxygen and the pH microsensors are used a lot. The first example shows oxygen measurements in brain slices. And in this paper here, the authors used oxygen produced from algae photosynthesis to oxygenate hypoxic brain tissue. So the oxygen sensor was used here to look at the oxygen production from algae, and they optimized the distance of the light. And they then submerged brain slices into this algae culture, and they used the oxygen produced from the algae to keep the brain slices alive. And they checked this by doing microsensor measurements inside of the brain tissue. And they found that the oxygen produced from the algae was enough to keep the brain tissue alive. Then we moved to an in vivo study measuring pH and oxygen in the brain of living rats. Severe birth asphyxia is the main cause of disability and mortality of newborns. This implies a decrease in systemic oxygen and an increase of CO2. So the authors here wanted to examine the effect of birth asphyxia on the brain pH and oxygen responses. They used a small tip oxygen sensor here and a pH microelectrode, and they implanted them both in the brain tissue and in the body tissue of rats. They induced asphyxia by letting the rats inhale a gas mixture of 5 to 9% oxygen and 20% CO2 in repetitive steps. And they implanted the sensors in the brain and in the body tissue, and they could then detect a low pH as a sign of acidosis, and they could also see a decrease of oxygen. That was again recovered when they increased the oxygen levels again. Yeah, so you could really use the microsensors here to describe these detailed responses of pH and oxygen when they induced asphyxia in the rats. Okay, so now to a very different research area, renewable energy. We see huge interest for using microsensors within this field, especially for the real-time quantification of hydrogen production using our hydrogen microsensor. And I will just go through a few examples to show you how this is done. The first example is about photoelectrochemical water splitting, where solar energy is used to convert water into hydrogen and oxygen at the surface of a semiconductor material. The authors here used a low-range hydrogen sensor to detect nanomolar concentrations of hydrogen evolved from the light during water splitting. And they used this new type of semiconductor material called a covalent organic framework as a photoelectrode. And during light illumination, the generated charge reduced water in the cuvette, and hydrogen was then produced, and they could detect it continuously here with the hydrogen microsensor. So you can here see when they turn on the light, the hydrogen started to be produced. Just another example here, it's a different tandem photoelectrochemical cell. And the authors here, they used both a hydrogen sensor and an oxygen sensor to measure the hydrogen and the oxygen produced here in the presence of light. So you can both measure hydrogen and oxygen here. Okay, hydrogen is the hydrogen sensor is also used in many biogas upgrading applications where microorganisms convert hydrogen and CO2 into methane. And in this environment, hydrogen sulfide is also present, and hydrogen sulfide interferes with the signal of our standard hydrogen microsensor. But we have developed a sensor that can tolerate hydrogen sulfide, and this sensor is called H2X. The H2X sensor here has a front guard that simply removes hydrogen sulfide in solution so that it doesn't interfere with the hydrogen measurement. On the photo here, you can see this H2X sensor mounted in a biogas reactor, detecting hydrogen in a hydrogen sulfide containing environment. And the hydrogen sensor here, it actually measured for 3 months at 55 degrees, giving a stable signal all the time. The hydrogen sensor here on the picture was mounted in a steel tube, and the sensor could then be mounted in the reactor with a switch lock fitting. On our website, we have a papers and references database where you can search for papers within your research field. So you can find the papers and the examples that are relevant for you. So this was the end of the presentation, and now it's time for your questions. Thank you very much for your attention. Please ask your questions in the chat, or feel free to unmute yourself and ask your question. Thank you. Protons, I'm not sure if are you referring? So the hydrogen sensor measures the partial pressure of hydrogen gas, not the protons. Please elaborate your question if I am not getting it right. So there's a question about how to stabilize small samples, such as grass leaf blades. Yeah, it's really about getting the sample very stable. You can use needles or whatever you have in the lab to fix your sample as best as possible, so it doesn't move when the pH electrode enters. I don't know if you have any comments on this.
It's often a good idea to place a small sample on agar, maybe, so then
there's
on
not
a
hard
surface. So there's not anything hard beneath it. You can go through it.
Yeah, so our sensors only detects hydrogen.
Yeah, so the hydrogen sensor detects the molecular H2 hydrogen. The pH electrode, on the other hand, detects the protons.
Yeah.
You can use that if you want to know that.
So the question about the best sensor to choose of oxygen to do a profile in sediments. Yeah, it really depends on the type of sediment. But typically, yeah, people use a size of 50 or 100 microns in sediments, but it really depends on if you have larger particles, stones in it. It also depends on the resolution that you would like to get from the profile, how steep the gradients are that you have in the sediment. But you're welcome to send us an email with some more information about the sediment that you are going to measure, and then we can comment a bit more on it. Okay, so pH, yeah. So pH is detected with the pH microelectrode. Yeah, there's concern about using agar, that it affect the recorded data.
Of course it might do.
Okay.
Yeah, so it might be that you want to just fix a leaf. If you want to measure inside a leaf, just fix it with some clamps and not have anything behind it. That's of course an option.
No, so that's a question about measuring in the gas phase, and the sensor measures both in the gas phase and in liquid, so dissolved gas in liquid. So you can use the sensor in gas phase and you can use it in liquid, just as you would like to. If the two phases of liquid and gas, if they are in equilibrium, the signal of the sensor will be almost the same. But for all the sensors, you can use them both in gas phase and in liquid.
So it's again the question of measuring pH on the surface of a leaf. Well, depends. You need to. You'd ask if you could put a drop of water on top of the leaf blade, and that's of course an option. You may of course dilute the pH, but in order to have a pH, you must have water, because that must be protons in solution. So there will not be a pH on the surface of a dry leaf, for example. There will be a pH just inside the leaf, where you have the juice of the leaf. So I'm not quite sure exactly what you get if you add a bit of water and then measure on top of the leaf.
Yes, it would be better to measure inside, but it of course depends on the leaf thickness.
Yeah.
If it's possible or not.
Yeah. So
yeah. You're welcome to send us some pictures and some more details about your experiment, and we can have a look at it. Yeah. Lifetime of the field data loggers.
Well, the lifetime of the field data logger is long, so there's a 1-year warranty. But I guess you may be asking for the battery life.
Oh, the battery life, yeah.
that depends on the instrument. So the field data logger mini, that has battery for about 20 days of operation at 25 degrees temperature. The field data logger, so the bigger one that operates all the in situ instruments, that has 20 hours of battery life, and we can add an extra 40 with an external battery. But we can also, if you're not more than 70 meters away, we can add a cable, so you can actually supply power. You can supply power from a bigger battery if you want.
Okay. Well, if not, then you are of course always welcome to send us an email if you think of something after this. And then we will just thank you for participating today,